My name is Ryan Lynch, and I'm a cannabis plant biologist. I currently work at the Salk Institute in San Diego. Prior to that, I worked as a commercial hemp breeder, and I was originally educated at the University of Colorado, where I did a PhD in the ecology and evolutionary biology department.
Excellent. So in your, I'm going to ask you some of the basics of cannabis genetics. And I know you've come out to Boston before and done a seminar with us on cannabis genetics. And I know these questions always come up, but if you wouldn't mind, can you please go over them again? There's this classic question out there in cannabis genetics having to do with indica and sativa. Can you please describe in reality the indica sativa differentiation and whether there is one?
Yeah, so there is no concise, simple answer. So I'll start maybe at the beginning, the origin of those terms. So indica is a Latin term for of India, as in like the Indian subcontinent, and sativa is a Latin term for cultivated. So these two terms, so cannabis sativa and cannabis indica, originated as species designations back actually in the 1700s. And a lot's obviously happened since the 1700s. But if we fast forward to today, now we find that basically all cannabis products, concentrates, flower, plant, whatever, has some type of sativa or indica description, or possibly uses the hybrid term, which means some kind of blend between the two. So we can look at it scientifically from a couple different ways. The first question is whether there is actually a chemical difference between products or plants that are labeled as indica sativa. And so a number of studies have addressed that through the years by going around and sampling dispensary products, for example, or in some cases doing more detailed chemical analytics of plants that have really strong associations with the term sativa indica. And honestly, the results just aren't entirely conclusive. On average, there are some differences in terms of particularly the terpene and terpenoid profiles between indica and sativa. But that doesn't necessarily mean that every sample that's labeled in a given dispensary is going to be necessarily different from one another. So it's really sort of an ongoing scientific inquiry into what these chemical differences are.
So originally, though, the indica and sativa designations were based on where the plant was found or physical characteristics or both, right?
Yes, so both. So they were used to describe plants that look different. So they weren't testing the chemical composition of the plants back then, but different cannabis plants have vastly different appearances, particularly when you're comparing, say, for example, fiber type hemp plants, which are also cannabis sativa, but look drastically different from what we see grown in a typical cultivation facility today. And so, the original descriptors were where the plants were from, and also the use of the plants. So one was supposedly more for a drug type usage, and that would be the indica, and then the sativa was more for like a hemp type accession.
Which makes sense because sativa is much taller, like in the common knowledge, right? The tall, skinny sativa, which having grown some hemp, that Prince of Persia that you helped me out with a while back, is so much taller. I mean, I think that plant got to like 16 or 17 feet. And then the indicas are meant to be short and shrubbier. Is that correct?
Yeah, that's the common vernacular use of the terms. Now, I can't really say whether the plants looked exactly like that in the 1700s. There are drawings in the original publications, so there are some things we can go off on there. But I think it's safe to say that given what's happened in history between then and now, that we aren't really looking at the same designations at this point. So I think that although the terms originated in these botanical taxonomic senses, the usage today with whether it's sort of an association with a sedative effect or a more energetic effect for sativa and indica, or the overall shape of the plant doesn't necessarily relate to those original descriptors. So, and terms can have different meanings through time. So there's nothing wrong with that. So I will add that the other way to look at it. Oh, go ahead.
Go ahead.
So I was going to say, aside from classifying plants based on their chemical composition, like testing for levels of various terpenes and cannabinoids, you can also look at the DNA sequence of different plants. And so other studies that have been conducted through the last 10 years have also examined the indica versus sativa question in terms of DNA sequencing. And in that case, also the results are not entirely clear and concise. There are different groups, sort of populations of cannabis plants that have been sampled, but they don't necessarily align perfectly to how cultivators and breeders describe them as being indica and sativa.
So, and that makes a lot of sense. We've been trying as a dispensary, as Seed, to help our purveyors, help our customers explain, indica sativa in terms of terpene profile, which I know isn't exactly correct, but at least can account for that more sedative effect of indica and more, active and energetic effects associated with sativa plants. But it's been very difficult to get the consumer away from these designations, because I don't know if it's psychosomatic or actual terpenoid profile related. But do you think that's the correct way to educate our purveyors, or should we be, working on a different angle?
That's a good question. I don't think there's a perfect answer for that. It just, in my experience, it seems like the terms are kind of there to stay. So anything that we can do to help make them more evidence-based and useful for the consumer, I think is a benefit. There have been various proposals over the years to replace the names, but it's been difficult because it's so baked into the culture, I think, at this point. So yeah, I think just providing as much data as you can in terms of terpenoid profiles is probably the best thing that you can do as a producer.
Great. We're on the right track. So the next thing I want to talk to you about is, you've been gracious enough to provide the museum with this array that I saw from you almost, I guess it was 8 years ago, in DC, when I first met you, and you were explaining this genetics array, which I know you've worked on since. We're going to have this on display at Seed to Soul in the Portland, Maine Museum. Can you walk people through what this means? It's a lot of information in a small picture, and I really want people to understand what it means and all the data that you collected to create it.
Yeah, for sure. I do need to pull it up though.
Yes, of course. Take your time. Gloria has it too, although if she shares it, we can't see you. So I better.
I just downloaded it from my email. So yeah, just
Okay.
1 second. Okay, so which one? Yeah, so I guess first thing is like, which one do you want to talk about? I can't.
The last one you sent me, I think, was the most up to date. Gloria,
I can look for it if you want to. Just give me a second. No.
Yeah.
You found it? Okay, give me a second if you want. Okay.
Ryan, what are the medals for on your door?
Oh, those are race medals from bike racing.
Oh, yeah.
Trophy room.
Trophy room. What does Poppy call it? The bang room.
I've never heard that.
Yeah.
Oh my gosh, why can I not? I made this. I should be able to find it.
Don't worry, I've been working on this for the last 3 weeks. I should be able to explain it to you. I can't find it either.
I mean, it's like literally right here. Oh, yeah, it's okay. Microsoft problem here. Hold on.
No worries. Take your time. That's what editing's for.
Here we go. This will work. Yeah.
You found it?
You just I'm on the way there. I figured out what I was doing.
Okay. I'm looking for it too, and I know I have it. It's just
it should be in the slides, Flo.
Yeah. Thank you. That's what it. But it's just so small. Trying to look for the big. Yeah, but here genetics.
Oh my gosh.
Genetics. What are you? Genetic.
I don't know.
It's okay. I can do it.
I found it. Okay. Oh, it's so small. Okay. Give me a second.
I have it. This email is just not a good thing to search.
Found it, but it was
Google to Microsoft. It's just been.
You can. I can share my screen for a second. You can barely see it, the small one, but let me just see. So like.
Sending it to both of you. Oh, it's unable to attach. Why is all my things unable to attach?
Too big. Yeah, another Microsoft problem that doesn't automatically.
I mean, I know that you can barely see it, so probably this is not going to be helpful at all. But
okay, yeah, just one second. I'll just dig up the original.
Yeah, that's the one that I have, and here, and I can find the original too. That's the one.
I just text you the message failed to send. Why?
Mine too. It's been happening to me all day, just so you know. And I thought it was my phone. Technology.
Oh, do you want? So yeah, which one do you want to look at? Okay, so I was looking at the. Do you want to look at this old one or what's the? Because I sent you that the other genome alignment one.
Yeah, the new one. The one I have is the one you sent me. Hold on, I'm trying to forward the email. The one you sent me has a bunch of
It's the one that I just got. And you saw that one. Look for it again here.
Figures. So I see one that says narrow leaf drug type. Okay, broadleaf. I have 4 figures. The one that's colorful is the one we were going to use.
Yeah, that's what I'm thinking. Okay.
That's the new one.
Is it? What's the file name? Is it?
The file is named GBS_WGS3_02102015.
Okay, so you don't want to go over this other the TWSVA.
I mean, I'll take whatever's the newest, whichever one you want to talk about.
Yeah, that's the newest. Well, they're just really different things. So so that's like a really old one.
All right.
Yeah,
well,
it's sort of
we didn't know
broader
which is which. So walk us through all of them, and then we can use both.
Okay.
Let me put it again.
Yeah.
Well, can I share my screen here?
Yeah.
Yes, please do. And you can share this on the right-hand side on the top next to leave, next to the red button. You see like the little one that says share.
I have never used meetings here, whatever this is. Stop spotlighting me. That's the one I see. Show.
Let me see if I stop spotlight. Let's see if that would change the. It should look. Actually, let me look. I'm gonna send you a picture and see if you see it.
Okay.
Not on myself.
If you press the 3 little dots, it'll say share.
Yeah, I don't actually have.
You don't have the share one?
Yeah, share is not an option for some reason.
That's interesting. I'm trying to see if I can give you. Okay, I won't pin you. Let's see if that make any difference. Did that make any difference in your screen? In the right, it should be like in the top on the right hand side. It's a little square with an arrow and it says share. No yet. Do you want to send it over and I can share it?
Okay, sure. Yeah. So let's see
I just
here.
I just replied all to the email as well. I don't know if you guys can get that.
All right.
Yeah, because it's important for us to know which one to use.
Yeah, absolutely.
Yeah. Which
All
one?
look
Yeah, we
similar
want
to us.
we want it to match the.
Exactly.
Did you send an email or something?
Email.
Haven't received anything yet.
No.
One of my clients will say, "Is Mercury in retrograde?" And that's why everything is not working right now.
Well, then it's been that for the last ever how many months.
Yeah.
Yeah, I just sent it, Brian Lynch. I'll send again, Gloria.
Yeah. Sometimes just a little lag.
If you want to send the email, maybe I can share it here, Ryan. Want to like send it to me, Gloria at seedyourhead.com. I can just share the screen.
Okay. Yeah.
Maybe that will help.
Okay. Okay, new email. Yeah, I'll just do that. Seedyourhead.com?
Yes.
Yeah. You got iPhone charger? Nah,
All right, so that one there.
Oh, cat in a bag. That cat looks miserable.
Poor thing.
Look, go ahead. Show cat in a bag.
Cat in bed.
Let me see. Hold on. I can see. Oh my goodness. So cute.
That cat looks super unhappy, Ram.
Poor thing.
Look who it is, Ram. It's Ryan. Well, you can't see him now.
Hey.
You remember Ryan? Remember we went up to where did we go? Colorado,
Rocky Mountain National Park.
Rocky Mountain National Park. Honey, get that cat out of the bag. He's miserable.
Cat in a bag.
I haven't received it yet.
Okay, I just like nothing's working. Okay.
Maybe a text message. Maybe you can just send a text message.
I tried it by
Try.
text. It wouldn't go.
Oh, I don't.
Maybe
yeah,
Ryan can send it to us.
I don't know how to put it on my phone. It's not on my phone. I don't really use my
That's in your
phone.
phone. Okay. Maybe a picture of it. Like if you take a picture
But
of the
Gloria,
graphic.
you didn't get my email?
Mm-mm. I didn't get your email. I didn't get your email. I didn't get anybody's email.
Oh, I got yours. Okay, yours just came through April. Yeah.
Oh,
Then
good.
now you find the figures. It has all 4 figures.
Yeah, those are good. Yeah, so yeah, we just use these are I see 2, but those are 2 good ones. So those are the main.
Oh, I finally got it. Okay, so
Yeah, I guess first question is what do you guys want to talk about, because they're 2 very different things.
Well, let's explain what they are, because we don't really know what we're looking at, and I want to make sure everybody else knows.
So which one will be the first one? That way I just, which one you're gonna talk first? What's the
So
name
I'll
of
talk
it?
about the this one doesn't have any colors in it though. Darn it.
Doesn't have the
But I know
4?
that I have the, I mean, I know, hold on a second. Okay, I have your email here. You send it to me. Now I have it, I downloaded it, put it in the. Okay, I have this one. Here we go. I've actually, give me a second, let me download this.
Sorry, Ryan.
Sorry, Ryan.
That's okay. Yeah, no, there's a lot of file.
Okay, so
Man.
share.
But I'm glad we're doing this because we didn't really know what we were looking at. It looks awesome.
Yeah, well,
But when I
yeah,
saw
we'll
you
talk through.
present it,
Okay,
it was
so
like,
yeah, let's just chat about this one first. Well, let's just decide, like, I mean,
oh, good, you've
I guess-
got it, Gloria. Okay.
So
let
we
me mute.
have that one, and then we have this one. I want to know which one that I don't know.
Well, that
the
one
thing.
is just a non-colorized version, and then then there's a TSV. There's another one that April just did.
Okay, so then which one do you prefer?
Well, no, there's another. Well, so there. Okay, so that is the best
there's
one out
two.
of these types, but there's
Do we
another
want to
trick.
discuss this colored one?
Oh, it's
and then there's
another one.
Yeah.
It's a better one.
No, yeah, no, I'm saying, like, okay, so okay, look at April's other, like, her email that she just sent
Yep.
at 3:26. So in that one, there's a file called
The 2015 or the TWSA?
TWSV or whatever it is.
Okay.
Yeah, that one. So that's just like a totally different.
Yeah. So
Yeah.
which
Okay.
one?
So this and the colored one is what we want, right, Ryan?
Yeah, that's what you'd want to.
Okay, so this one, and let me open the other one very quick. So that we have, and this one.
Yeah, those are probably the best
Okay,
two.
perfect. And okay, so let me open show and folder. There's another one. Sorry, wrong one. It's just the order. So we have which one do you want to open? Where first, the TWSVA
Yeah,
or the other? That one. Okay, perfect. Let me open.
Okay.
it
So once you
now.
open that, I'm gonna mute because I have stupid traffic out my windows. But then Ryan, I'm just hoping that you can just describe these because we're gonna put them up, and I know there's a lot of valuable stuff in there, and that's what I want to get.
Okay, sounds good. So should I just go, I guess?
Which one? What do you think makes more sense first?
Well, we'll just do you. I mean, edit it later. Yeah, we'll just start on this one. So, okay, so this is a whole genome alignment of fully chromosome assembled cannabis genomes from two very different types of cannabis. One is train wreck, and the other is called herb. So they're both type 1 cannabis that makes THC, but aside from that, there's not many other similarities. So in these type of plots, you can see the chromosome tracks are represented as rows. And so for each row, like on the top where you have chromosome 1, you have an alignment between the train wreck and the herb chromosome 1s. And then the coloring between the two chromosomes tells you about where the similarities and the differences are. So if you go from left to right across chromosome 1, you can see the gray area, and that represents what we call a synthetic part of the chromosome, meaning that the gene sequences are the same between the two samples, but as you go along, you start to see some different colors. Where you have white areas, that means that the two genomes are so distinct that there's really no way to make an alignment between them. Where you have green areas, those are called translocations, and that means that some of the genetic information from one of the chromosomes is found in a very different location in the other chromosome. Duplications are coded in blue, and those are areas of one genome that have been copied multiple times and are represented at a higher copy number in the other genome. And then on this chromosome 1, probably the biggest feature here is in orange, and that's called an inversion. So across that entire area that you see in the orange hourglass shape, you have a completely inverted sequence when you compare the two samples. So in this case, that's over 25 million different base pairs, and there's hundreds of different genes within those inversions. These have never been documented before in cannabis at this level. So this unpublished work is still endeavoring to determine the types of genes and what effect those might have on the phenotypes of the plants.
And so how did you select the chromosomes that you evaluated?
So this is part of a larger study. We have about 170 total cannabis samples included in what's going to be one publication. So in this case, I'm just highlighting two of the very distinct different types of samples that we have.
And what does the inversion mean to you? So like using chromosome 1, for example, the inversion means, you know, it's the exact opposite sort of arrangement of, you know, adenine, thymine, guanine, and cytosine. Like, is that what I'm?
Yeah, that's right. So yeah, it's just the letters that have been reversed. It's a type of mutation, and particularly in plant genetics, it's been difficult due to the cost and technological limitations to catalog those. So most plant genetic studies up until recently rely on smaller mutations, for example, like insertions or deletions, or most famously single nucleotide polymorphisms. SNPs, you hear them called sometimes, and those are just single base pairs across the genetic alphabet where you have a single difference. So in this case, with that inversion, you have 25 million different base pairs that are in a different orientation. And so while within those inversions, there are genes that, you know, presumably have effectively the same function, they're just in a different orientation. It does have a lot of implications for breeding and making new types of cannabis. When you have plants that try to reproduce that have inverted regions like that, you can't get crossing over. So exchange of genetic materials is limited, and that can cause issues with the offspring. So it's a very interesting and different type of mutation that has been documented in cannabis before.
Okay, well, that's fascinating. Ryan, would you mind turning your camera on again in case you want to use it elsewhere?
Oh, it is. Yeah, it should be on.
Yeah, I can see him.
Oh, you can? Okay, I can't, but it doesn't matter. All right, good. As long as Gloria can.
Yeah.
Okay, so I'm going to go on a little tangent here because a long time ago, I watched this BBC documentary about genetics called The Code. And it was so interesting because it resulted in what I would name my band if I had a band, which is Amplified Worm. Because what it talked about was the fact that our human DNA shows basically worm DNA, but repeated over and over and over again, and not in like.
We can hear you, April.
Oh, she cut out. Uh-oh.
Can you hear us? Okay,
Yeah.
now we can hear you. Yeah, you're good now.
Okay, good. So it showed these steps of genetic, you know, evolution where we are going up quickly by amplifying, meaning like repeating the worm DNA over and over again. And I always thought about it, and I always think about it, and I don't know if this is relevant at all. It's more of like the philosophical parts of, you know, genetics. But what explains these inversions? Are there any hypotheses out there as to what is explaining these inversions as opposed to like the single mutations that we see?
Well, there's different mutational, you know, all mutations, all differences that exist in life today originated from technically what we call an accident. So mutations aren't something that are, you know, like pre-designed. They just happen. They're actually you can think of them as mistakes. So the inversions happen when there's a couple different mechanisms by which they happen, but a lot of times it's really just that the string of DNA, when it's being replicated, actually breaks, and then there are enzymes actually that go around and fix those kind of things, but sometimes they don't fix them correctly. So if you can imagine a big chain of, like, you know, strings on a bead, for example, and there was a break somewhere in the string, but then you put them back together and tied the string back together, there could be some misordering. So it can be as, you know, as simple as that. But the implications for
So
how
they're-
that affects the offspring are, you know, much different.
So they're almost getting read backward. You know, they're crazy.
Yeah, that's
Okay.
one way it can happen too. So there's enzymes that go around, and when DNA has to be replicated, like when you're making a sperm or an egg cell, or just a new cell for like a new, you know, part of a leaf or something, there are enzymes and different proteins that are involved in that process. And yeah, as you can imagine, they are error prone, and they do make mistakes sometimes. But the way evolution works, it turns out that, you know, on occasion, most of those mutations don't have any effect. Some of them are actually really bad, of course, and can be lethal or not adaptive. But, you know, in some rare instances, those mistakes can be beneficial for the offspring in the long run. And those are the ones that tend to propagate through time. So that's the simple explanation of evolution.
Okay, thanks. I needed that on this cannabis talk.
Very
So-
quick before your next question, April, sorry.
Yeah.
We had the other graphic. I don't know if we were gonna explain it or no. I just want to make sure that we don't forget.
First, I wanted to go back to that graphic, the one we had. I don't know if you have it up, Gloria. Apparently,
Yep.
I can't see the same things you guys see. But is there anything else on this graphic that we need to talk about or explain?
I would just highlight that, you know, due to the ability to sequence and assemble these genomes at this type of resolution, which has never been seen before, it's really answering some questions about cannabis, but it's actually reopening some old questions, including whether or not there may have, at least at one point, been multiple species within the genus cannabis, because we're finding so many differences that haven't been documented before. You know, in the last 20 years, most of the scientific literature has concluded more or less that all cannabis plants, whether it's, you know, a 20-foot tall European fiber hemp or some rustic Asian hemp plant, or, you know, the best marijuana from some dispensary in Boston, are really all part of the same species, Cannabis sativa. And part of the rationale for that is that you can interbreed all those different types of cannabis and make viable offspring. But that isn't, you know, the strict definition of the species. So, you know, now we're finding that with these larger scale, what we call structural variations, like the inversions and some of the translocations, that these may be relics of former species, which may or may not really exist in nature anymore at this point.
Okay, that's super fascinating and gets me to, well, I guess first let's go over the second graph, the second array, because I want to then talk about some of the where we are today with genetic variation. But first, I'd like to go over this sort of starburst array and
Okay, cool.
have you walk us through what this means.
All right, so this is a phylogenetic network analysis of cannabis samples from all over North America, as well as some samples from Europe and Asia. This was part of my PhD dissertation work and was published back in 2016. So it's based on DNA sequencing and assessment of the single nucleotide polymorphisms that are assessed by aligning reads from each sample to a single assembled reference genome, which was called Purple Kush. And basically what it does is tells us that there has been a lot of hybridization, a lot of intercrossing between, in particular, North American drug type cannabis. So in this graph, you can zoom in and read all the different sample names, some of which will be familiar, classics from dispensaries through the years. And the way to read these graphs is that you basically can measure the actual genetic difference between any two samples by going from one tip where the label is, and then following the line inward until it meets a node that then you kind of reverse direction or follow it in a different direction to another sample. So there's actually a scale bar on the bottom right-hand corner. So it's actually a quantitative map of genetic distance between different samples from, I think, about 200 samples at the time this was published. So you can see some structuring to it, although it sort of looks kind of like a cloudburst, but there is some separation, particularly on about 3 or 4 o'clock. You can see a group labeled hemp that has some larger gaps compared to the other parts of the graph. And so by hemp, in that case, what we're talking about are European fiber and hemp seed type hemp, rather than the high CBD yielding hemp, which actually there are samples from that as well in this analysis, but those are actually much more closely related to the type of marijuana plants that produce THC than they are to the European fiber hemp. So the big take homes from this are that there does seem to be a little bit of structuring, differentiation between drug type groups, what we call marijuana in North America. So there's labeled at about 1 or 2 o'clock the broad leaf drug type. And so you can roughly equate some of those samples to what people sometimes call indica. And then pretty much the rest of the wheel, excluding the hemp, are more mixed or narrow leaflet drug type samples. So yeah, there's been obviously a lot of hybridization and crossing in cannabis, particularly with the drug type. And then it seems like hemp has actually had a very, European hemp has had a very limited role in breeding of North American drug types.
How did you select the plant, the cultivar that you compared all these to?
Oh, so that was another, yeah. So that was the genome assembly that we used to align all the smaller reads from each sample to, that actually was what's called a reference genome that was published by another group in based out of Canada.
Oh.
So for most plants, up until recently, there would be like one reference that all the different labs use, or maybe a couple. But now, as genome sequencing technology has moved, just light years ahead, just in the last 5 years, now we're getting to the level where it's like almost every sample that we're processing as a genome lab could almost be a reference. So it's completely changed the way that plant biology is done.
Oh, that's super interesting. So, Ryan, this part of what the museum is trying to understand and express is what prohibition has done, first to the people, which, you've been to the Boston location, but also to the plant itself and the genetic and chemical composition of the plant. From your research and, the data that's available to us, can you opine on like the impacts prohibition has had on the genetic and chemical makeup of the plants?
Sure. So I think that's one of the really outstanding open questions, and something that I actively think about on a regular basis. And we don't really know where the plant would be. Like if you've contemplated the counterfactual of their prohibition just didn't happen for cannabis, the plant would just be a completely different thing. And it's hard to know exactly what route the species would have taken, and where humanity would be for that matter. But looking backwards from where we're at now, it's interesting. It does seem like some areas of the genome have lost genetic diversity, or at least they don't have much at present right now. One of the issues with answering this question is that we just don't have samples from cannabis before prohibition, which is about 100 years old. Now, in most plant species, you actually would have access to seeds that have been preserved and maintained that are well over 100 years old. I work also on soybeans, for example, and you can order seeds that were collected from wild places all over Asia for soy over 100 years ago. And it's not the same seeds, they regenerate them every few years, but they do this for like thousands and thousands of accessions for every species. But one species that they don't do that for in the US based gen bank is cannabis. They just started accepting new accessions within the last 2 years, actually. And so there's fewer than 10 accessions, and none of them really have that much historical relevance. So so that does present some challenges for answering the type of question you've asked. One interesting way to go about it, and a method that I'm using, is to use feral hemp. So feral hemp are plants that used to be cultivated. They're descendants of plants that were originally planted for hemp crops at some point in history. We're not actually sure where, but the seeds, basically escaped from the farmer's field and just, continued to propagate. So all over, particularly in the Midwestern U.S., centered around Iowa and Kansas and Nebraska, there's large populations of these plants, sometimes called ditchweed. And those are at least 80 years old, because, there was no hemp between, the 1940s and 2014 in the U.S. So they've been out there quite a long time in isolation and also adapting to the Midwestern climate. So I look at those as, to some extent, a little bit of a time capsule that we can use to answer these questions. But yeah, to answer your question a little more succinctly, it seems like there's been some loss of genetic diversity in cannabis. At certain genes, particularly like the cannabinoid synthases. So those are the actual genes that make THC or CBD or CBG. In my analyses recently, it appears like there just isn't a lot of genetic diversity left in those genes. Now, other parts of the genome display extremely high levels of genetic variation. So we're kind of still trying to sort through how to interpret that, but yeah, it's interesting nonetheless, and I think we'll hopefully, using genomics and other approaches, like for example going through museum samples. So there are herbaria, so there are collections of dried cannabis leaf available all over the world for researchers to access. And so the problem with those is that the DNA is often quite degraded. It doesn't store very well for long periods of time, but there are some researchers that specialize in utilizing those to kind of piece together some of the parts of the puzzle to the question that you just asked. And I'm really hopeful that in the next, over the next years, we're going to really unravel a lot more about that.
Do you think that, many years ago when I first got into this, and this story, I was working with Dr. Lyle Craker, who has since passed and was a really amazing advocate. I actually just found out he passed. Really big advocate, really amazing scientist, really interested in the cannabis plant. And he had connected me with a person in Iran who was able to ship me seeds that were deemed medicinal hemp. And over a course of events, ended up popping a bunch of those and only got males, and ultimately crossed those with 4 just great breeds, Humboldt Sour D, Lilac City, Gorilla Glue, and one more that I can't recall. But at any rate, so I have this cross. Those plants grew to like 15 feet, had some interesting compounds in them. But the point is, do these seeds exist in other countries around the world? Do you think it's futile to run around and try to chase these landrace? I mean, but for the political issues involved in some of these places, could we find some of these, older genetics just growing?
Yeah, it's definitely worth doing. I mean, it's important to try to save the genetic diversity that's out there, and there are people pursuing that. There's going to be private collections. There's some countries have some more open germplasm preservation, but Yeah, I think most of it's going to be either outgrowing in the wild or in private collections. And it is worth, yeah, I mean, it's a long road to travel to identify and find pockets of diversity and then confirm them, because you have the other issue where seed companies have been selling elite sort of performing high yielding THC cultivars online for decades now. I think since the mid 90s at least, right? And so these seeds have made their way backwards too. So whereas a lot of people associate traditional landrace growing areas such as Jamaica, Malawi, certain parts of Africa, the Caribbean, and that is true. The growers there haven't just sat around either. They've been trying to advance their yields as well. So, in a lot of cases, we think that a lot of the original or more original germplasm and landrace material has been sort of almost contaminated with more high yielding plants. But there's ways to test that too. So, yeah, a really refined approach would be target the right areas, and then go in there, try to sample the plants correctly and ethically, give credit where credit's due, which a lot of people don't do, obviously, and then really try to confirm using genetic tests what actually the origin of the plants that are supposed landrace material are.
So if you could pick 3 places that you'd like to go to hunt these, I remember there was that show. Was that
Strain Hunters with Arjun.
Hunters. Yeah.
Yeah.
One of those guys recently passed as well. I think he succumbed to something. That's a shame.
Malaria, actually. Yeah, Franco got malaria in Malawi.
Yes, exactly. But that was a cool idea to go hunt down these strains. If you could go, as a scientist hunting these landrace to a couple places in the world, where would you pick, barring any sort of political or dangerous situation?
Well, the latest research indicates an unusual area that strain hunters didn't travel to, which would be northern and sort of eastern China, as being really the center of diversity of cannabis. So they're not going to be landrace, sort of classic marijuana landrace types, but the genetic studies show that that's really where the most genetic diversity is. But no studies have really gone in and cataloged some of the classic hotspots that, for example, strain hunters highlighted. So, like, I really haven't seen any good studies on, Jamaican land races or comprehensive studies of Africa or South America. So it's still really an open question. I mean, I'm working on some South American samples that have some very interesting traits. For example, a lot of people may have heard of auto-flowering plants, which are plants that are daylight insensitive, meaning that they basically start flowering within the first 4 or 5 weeks of their life. And that's a trait that usually is something that naturally adapted to very northern climates. South American land races that I've worked with show really the opposite phenotype, where even if you were to try to induce flower using a normal cannabis cultivation protocol, which is 12 hours of light, 12 hours a day per day, they actually won't flower, at least upwards of 6-7 weeks. So, and that makes sense because in the tropics, it's basically always about 12-12. So, plants aren't going to get very big if they're just flowering as soon as
even if you put them under 12-12, they're just going to take their time and do what they want.
Yeah, there's a whole gradation of phenotypes. So some of them they'll start flowering. You have to leave them in 12-12 for like 7 weeks, and then they'll start flowering. But I'm not sure if that's an interaction between light cycle and also root boundedness, which I also believe is, at least in some cannabis lineages, related to flowering. But yeah, there's other ones that we think can basically go almost indefinitely. at 12:12, and it's not clear what their flowering trigger would be in their natural environment. So.
Oh, that's amazing. Well, so we have so much to learn and apparently some traveling to do. My mind is now racing, and I will probably call you next from northern China. Or send you a ticket.
Yeah, or you could just invite me. Yeah.
That's what I was gonna say. I'll just send you a ticket. It's easier.
I have a passport, so you know.
That sounds perfect.